to talk to you and see how it goes from there. So I'm likely going to recruit one master student. So
Yes,
I
sir.
have found my PhD student, and I have started the paperwork for that student.
Uh,
Now I'm looking for another master student.
yes, sir.
So so there are a couple of candidates. Means I receive almost like 5 to 10 emails a day. It gets very overwhelming to find out the right fit for the program. Means they have a skill set, passion, and all those things, but you have to look for are they right fit for the program.
Yeah, sure.
So yeah. So how about these? We go ahead with our 20-minute presentation, and then we'll have a quick questions answers, and then we'll take it from there.
Sure, sir. Shall I proceed?
Yes. So, and would you be okay if I record the presentation?
Yeah, of course, sir.
Because
Okay.
that will help me to kind of review it later on.
Thank you, sir.
So, sure. So, just a second. Where is it?
So you have to give me permission for sharing slide, right?
Oh,
So,
sorry. I should have multiple participants. Now you should be able to share um,
okay,
your
perfect.
screen, your presentation.
I
Yes,
think,
I can see you're on your email interview presentation. So I will let you continue with your talk, and then I'll start. I'll mute myself.
yes, sir.
Um, okay. Okay. Just can you do the full screen?
Okay. Is it fine?
Yes. Perfect. That's perfect.
Okay. Let's start to my
Recording
present.
in progress.
Sure.
Okay. Okay. Good afternoon. Namaste. I am Vikas Vasmith, and I have done my graduate degree from Agriculture and Forestry University, and especially in agriculture science, right? And awarded it in December 27, 2023. Talking towards my the international credential evaluations in undergraduate degree, I have completed 165 credit hour course from 1 to 8 semester. Likewise, in undergraduate degree, I have got an opportunity to work with senior colleagues, professors, and some of the recent researcher. And I have published seven plus research documentations that is already available in the Elsevier. Some of them are Sailpraise, Hindwai, and one of them is Economical Frontiers. And 2 years Google Scholar citation is 26. Our university does not provide the verified institutional mail system in undergraduate courses, so the Google Scholar account is not verifiable. So beside that, that research documentations, I also publish a systematic review book that is available in the Amazon, especially the wheat rust, especially three domains, say the France, Germany, and USA. And currently 9 plus documents are under publication. Some of them are Springer, BMC, and one of them is scientific report, right? Beside my academic pursuit, I prefer to play the flute, and I have been playing the flute since 16 years. Let's talk about my work experience. And in undergraduate degree, especially in the internship period, I was assigned for Nepal Agricultural Research Council, and in internship period, I got an opportunity to more experience on some experimental designs and some lab experiments, say like the PCR. In internship period, I got more familiar on some PCR DNA extraction to the CTAB method, PCR, and we did the band scoring of Landes' maize varieties using the lambda marker, right? So beside that, we also did the field trial evaluation for the sport blots in rust for screening of more than 862 plus genotype for that pathogens, right? So, and in Maize Research Center, at that time, I got an opportunity for the pollination techniques for the hybridization purpose, and the scoring for spuring for the northern and southern blight of maize. And for the at that time there is a peak period of the fall armyworm, so we extract the trichoderma and do pure culture and implies for the fall armyworm. And here is my some glimpses that shows in undergraduate degree. So this is a greenhouse trial that is for the mung bean, and I aim to select the most climate resilient, temperature resilient genotype, heat stress resilient genotype for the mung bean. And this is the field experiment that is specially designed in augmented block design, and that experiment is for the analysis of post fertility as well as the drought stress tolerance line recommendations. Similarly, in maize research program that I got an the visualization of one phenomenon, one picture that is simply about the development of the seed on the silk. So our objective is not to solve this development of the seed on the silk, but I got at that time I reviewed many papers related to that, and one of the paper that described from the Barbara M.C. Nastak from the Cornell University. So I thought this is probably the effect of the jumping interactions, right? And this is the SPAD meter that measure the nitrogen contained in the leaves of this is the mung bean seedlings. This is the demonstration plot of the wheat we scored for the sport plots and lost for more than 862 lines of wheat. And this is simply the gel matrix, and there is some bands pattern that move for the negative to positive sides, right? And this is the pure culture techniques for the sport plots, mother culture for mother culture purpose. And here is the thesis dissertation that I have submitted to my university as an undergraduate degree, which was entitled as a genetic variability study blob modeling, especially for MTSI, multi-tiered stability index analysis for the stability as well as the best performance line identification, as well as for the post-fertility state evaluations. Here goes for my data analysis experience in my that I have acquired in my undergraduate life. So here is the correlations network graphics that I was drawn from the methane package of R, and this is the recent correlogram that I have created, and I'm going to launch a recent package for these types of plot, right? More aesthetic. And this is a circular representation of the bar diagram. If we have the multiple variables and multiple level of treatment, say the more than 30 15 30 50 40 at that time, circular repetitions of that particular variable, a particular treatment level is more precise and more aesthetic. So here it comes from the MTSI, multi-tiered stability index analysis techniques, that is used for the stability analysis, right? These red dot genotypes which are selected for the overall tape. This is the mixed model effect analysis, and that is and that is a random effect model, right? And this is the radar plot that shows the strength and weakness of the each genotype. And this is simply the box plot. These are some points are concentrated outside from maximum value, and this is the minimum value, this is mean value, this is q1, and this is q3. There is some data analysis techniques for I have learned from certain data set from NCBI tools. So this is the Manhattan plot, and there is a log value. More negative log value is more precise for particular SNP, right? So if we have multiple variables, multiple traits for particular phenotypes or particular SNP, so it is more accurate and more standard that's represented in circular form, right? One trait for one circle, second trait for second circle, so on. And this is the I have created this plot using CM plot package from the R, right? So this is the SNP density marking of from the 0 megabase pair of particular chromosome 2 the 3.2 megabase pair. This is also the data set I have taken from the NCBI supplementary files, right? And I have done the analysis again that. So I use the QQMan package to draw the SNP density marking, and here is the this is the circus plot, right? I have used 62 wheat genotype for the representation of the particular phenotypic marker, especially the LR34 that is associated with the sport blotch resistance genes and rust resistance genes. And here is the mixed model analysis techniques that I have developed, right? This is the integrated uh, plot, that is Poisson and box plot, and here is a certain error bar that depict the uh, error percentage of each data point that is concentrated on particular variable, particular genotype, or particular treatment level. So in mega environmental analysis, there is mostly used protocol is MTSI, BLOB, WAPS-Y, and AMI. So AMI and the GG biplot is generally used for the fixed effect model, and BLOB and MTSI used for the random effect model. But which one is more precise? So there is a need of the cross validation techniques, right? So this is the cross validation techniques for the fixed and random effect model selection for particular experiment, especially the replicated data, right? So this is the principal component biplot axis. These are the major two dimension. Here is one dimension one, here is another dimension two, and here is the biplot, and that this biplot contains certain genotype and associated certain phenotype, right? The phenotype simply means what we see and what we eat. It's called phenotype, and especially those marker or the phenotypic marker considered in a particular cluster, if they are more economical, we can select for further breeding program, right? So this is the circus plot. I have recently used the GFF file format, that is the general feature file format, and GTF, general gene transcripts file format, in the BSU core platform of the Pythons, and I have drawn certain positions from the particular location, starting positions in positions of a particular trait or particular SNP or particular exons, or maybe the particular genes or particular nucleotide, maybe upstream, downstream, right? Here is the also this is the heat map. This is the WAPS by indexing for the yield stability analysis. Stability simply tells about the ability of the genotype or varieties or cultivar that perform constant phenotype over the diverse environment, right? In quantitative genetics, there is one term that is called homeostasis. So we can resemble that terms with the stability, right? So stability means, let us take an example, say if a genotype have a ability to perform in worse environment is same as in the good environment, that is stability. That means give the constant phenotype, either is under nutrition conditions or stressful conditions or more more resourceful conditions. So this is more precise to detect the mixed effect model for the stability predictions, as well as the ideal performance candidate selection among the diverse populations, right? So this is the genotype environment interaction analysis for the mega environment, which environment is more precise, more favorable for particular genotypes. Here is environmental index. Here is the Sputnik 1 plus maize genotype. This paper is under publication in scientific report, right? So this is the dimension contribution factor analysis for the principal component analysis, right? Which principal component occupies the which trait, and how much percentage contribution by that dimension or that principal component, right? So and this is the threshold line, and there are several variables that meet the threshold line, that surpass the threshold lines. These are selected in the first principal component. Similar, the second principal component same follows the principles. And here is the scree plot that shows the cumulative percentage of variance explained by the each principal component and their respective LGN value. So more than those those dimensions which have, or those variables which have the more than one LGN value, that is selected for the analysis purpose and interpretation purpose, right? So here is the recent graph that I have done via core, and this is the chromosome 5, chromosome 6, then that is associated for the starting position in positions, and that is specific mark associated that trait. So more aesthetic representation of that plot. Here is 12 elements, right? 12 plus element, that is gene associated for particular two chromosome 5 and chromosome 6, and their respective positions in the chromosome, that particular locations of the chromosome. That is called QTL analysis. Simply the right. Here I learned from the one university conference. This is the linkage mapping, and this is the QTL distortion analysis. Those population which does not follow the random Mendelian inheritance patterns we generally use to analyze the QTL distortion analysis, or those markers, those SNP, which do not follow the random inheritance pattern of Mendelism. So this needs to filter out. So this is the recombinant fraction matrix calculations, and this is clustering of particular gene or SNP for particular chromosomes, right? So this is the parents one, and this is the parent two, and there is the associated trait SNP, or gene of interest, say, right? Their position from particular chromosome from centromere to telomere, that is centimorgan distance. This is the chance of recombinant frequency. If these two genes are too close together, that is the chance of linkage drag. If they are far from each other, that is chance of recombinations, high recombination frequency, right? And here is some these are the cluster analysis for the genotype. So there is a UPGMA method, hierarchical clustering method for their pedigree analysis, their specific trait analysis for clustering particular group. So this is the linear form, and this is more circular form, more static form. This is the height distance form, particular set. So this is the WAFs and blob plot, based linear unbiased prediction plot, that is used for the mix random effect model analysis. And here is some there is a genetic scientist centenary study of the plot of the 41 genotype, right? And this is the fourth analysis, especially the yield, and which variable directly contribute, which variable is indirectly contribute, and their coefficient value is represented here. So the question here is the why I need not only the molecular biology but also the computational genetics. So during my internship period or undergraduate degree, what I am able to conclude is that there is a significant gap between the research stations and farmer fields. So why are always farmer taking the burden of that, right? So from some literature review and from my experience on crop improvement programs, so I can say that the phenotype that we observe or that we eat is governed by the three factors, right? One is genotype, gene associated particular trait. Second one is the environment, and third one is interactions. Many developing countries like Nepal, Pakistan, Bhutan, they are concentrate their most of the research fund in the term of the crop improvement programs, but there is the significant gap. This is due to the lack of stability, right? So stability is governed by if we multiply the frequency of the desired gene from the genotypic level, molecular level. So there is an integrated approach, right? That's ultimately potential increase the up to the potential leap in the farmer field level. So I need molecular biology this way. So I thought in your expertise, I will get more familiar on the more molecular techniques, lab experience, say like PCR, CRISPR analysis techniques, marker-based analysis techniques, genome-wide association studies, some techniques for the gene of interest marking, especially the quantitative genetics. Like so, I am deeply committed to reduce the significant gap between the farmer fields and research station. So as a dictum, I have to I need this molecular biology, and capacity building and innovation exchange is a major theme of my molecular biology and computational genetics, right? So here is the final slide, right? That describes about my the journey from this point to this point. Certainly, I will be there if I got an opportunity to work with you. Thanks.
Recording stopped.
Thank you, Vikas. It was very elaborative. Overall, I was able to understand what you have done so far and what kind of skill set you are developing for your career development. One of the thing I want